Hello everyone, welcome to the class. Today we are going to discuss the topic cofactors under the chapter Biomolecules. So let's start with the same. We know enzymes are made up of proteins, tertiary proteins. It means the polypeptide chain arrange itself to form a globular structures. So enzymes are the globular proteins. Now, when they are participating in a reaction, sometimes enzymes themselves are sufficient to catalyze the reaction, and in few cases enzymes get associated with non-proteinaceous part to make the process of catalysis more efficient. These non-proteinaceous chemical compounds are termed as cofactors. So let's go ahead with the same. So enzyme can be simple enzyme made up of only polypeptide chain like pepsine, trypsin, they are simple enzymes. When we talk about conjugate enzyme, in conjugate enzyme there is one proteinaceous part and one non-proteinaceous part, and collectively we are going to use a term for that is holoenzyme. Fine. Holoenzyme is having two parts, the proteinaceous part, that is apoenzyme, and the non-protein part termed as cofactors. So the topic is cofactor. Cofactors, it means they are going to attach to the site of an enzyme where the substrates used to bind to make the process, to make the reaction to occur in a system in a proper manner, or we can say to catalyzing the reaction properly. So these are the cofactors. Cofactors can be further of three types: prosthetic, coenzyme and metal ion, or we can say metal activators. Now, how they are going to attach to an enzyme, where they are going to attach, what are the examples, we will discuss that. So let's take an example of this holoenzyme once. This is an enzyme. This is that active site of an enzyme, and one particular structure, for example at this particular part, this cofactor is going to attach. This cofactor can be prosthetic, can be coenzyme, can be metal ion. So this part is apoenzyme, and the whole is termed as holoenzyme. The whole is holoenzyme. Fine. Now let's talk about the prosthetic part. This prosthetic group is organic in nature, tightly bound to the enzyme. Okay, tightly bound to the enzyme, and it is permanently attached. It means this is always remain with the enzyme even after the completion of reaction. Okay, that will remain attached. Example of this is heme. Okay, you might have read about hemoglobin. So in that globular protein, heme is one part. In the same way, there is a catalase enzyme, there is a peroxidase enzyme in which heme act as a prosthetic group. Got it? So that will remain attached to the enzyme for catalyzing the process. Now moves to the next. Coenzyme is another type of cofactor. This is also organic in nature. The difference is what? Coenzymes are going to remain or attach to the apoenzyme loosely, and they attach only at the time when they have to participate in the reaction, and when they have participated, they will get detached from the apoenzyme. Okay, so they are loosely bound cofactors, okay, and bind only at the time of reaction. We can say catalysis के time पर ही attach होगा. Fine. Example, mostly the vitamins, they act as a coenzyme. Example, NAD, NADP, these are the enzymes, FMN all. Okay, so in this particular coenzyme, the vitamin niacin is there, which is B3 vitamin, act as a coenzyme for this particular apoenzyme. Fine. Metal ion. Metal ion, these are the metal activator. As they are the metal ions, so these are inorganic cofactors. So the examples like iron, copper, okay, manganese, zinc, in that way. So these ions, they are going to help in catalyzing the process of reaction, or we can say facilitate the binding of the substrate to the enzyme. Example of this one, like these are the metal ions. So if you are able to recall the digestion, there we have read about carboxypeptidase enzyme from that pancreatic juice that need this zinc ion. So there the zinc ion will help in catalyzing the reaction of this carboxypeptidase enzyme. Thank you.